In this video, we will discuss about the results where they check the effect of calreticulin on the EGF-induced EMT. So in the previous video, they have shown that they have made this calreticulin silenced Capan-2 AsPC-1 and also BXPC3 cell lines. Now they wanted to check whether they have this affect the EGF-induced EMT or not. So for the scramble control, they are able to see the spindle-like structures in the EGF-induced cells, and they have used the concentration of 50 nanogram per ml of EGF. Here we can see the spindle-like shape, more spindle-like shape compared to what they have seen in the scramble. And similarly, and this is for the Capan-2 cells, and similar results they have seen for AsPC-1, where they have seen that spindle-like shapes in the scrambles cells, that scrambles treated cells that are induced with EGF, and in the control they do not see any spindle-like shapes. However, for the calreticulin silence, that is SG1 CRT and SG2 CRT in the Capan-2, they also said in the text that they do not see too much spindle-like shape that they have seen in the scramble. And similarly, for the AsPC-1 also, it is much more clear that the SG1 CRT for the 1% BSA and 50 nanogram per ml EGF are almost similar to each other for both SG1 CRT and SG2 CRT. However, for this Capan-2, it does not look like similar, but signals looks to be less spindle compared to the scramble cells that are treated with EGF. The few suggestions that could help them to show that it does not induce EMT would be to check the EMT markers using qPCR, or they can also use immunofluorescence to check the EMT markers like bimantine and E-catherine. That would give like better results, and people would be easier. It will make the reader easier to convince that this calreticulin affect the EGF-induced EMT. And they have seen these similar results also for this BXPC3 cell lines, for which they have also made this calreticulin silenced stable cells. So far they are able to come to a point that calreticulin plays a role towards this EGF-induced epithelial to mesenchymal transition, and in the absence of calreticulin, those EMT, that is epithelial to mesenchymal, EGF-induced epithelial to mesenchymal transitions get affected, and because of that, it will also affect the cells aggressiveness, pancreatic cancer cells aggressiveness, as we have seen that as we have not seen, but researchers has published previously. This same group of researchers published previously that calreticulin is so high overexpression of calreticulin is associated with the aggressiveness of the pancreatic cancer. So they wanted to check whether they are checking whether this calreticulin has a direct role in this EMT or not, that is epithelial to mesenchymal transition, and they can see those effect for the 3 different pancreatic cancer cell lines that they have tested, that is Capan-2 AsPC-1 and also this BXPC3. And they have tested not just in the 1 CRT, it is in the 1 silenced CRT, but 2 silenced CRT for each cell lines. That is for Capan-2 AsPC-1. This could like from these results they finally come to the point that calreticulin has some role, and it might also affect the pancreatic cancer aggressiveness, or it might affect the pancreatic cancer's migration and metastasis ability, because it will affect this EMT, that is epithelial to mesenchymal transitions, and cells that are in mesenchymal state are more prone for this metastasis and migration. That's all for this video. If you need to understand more about these experiments in detail and wants to look the supplemental figures for BXPC3, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.